Hello everyone, my name is Chloe Faris, and today I'm going to be investigating the bacterial role in acid mine drainage passive remediation systems. There are over 5,000 contaminated watersheds in Pennsylvania alone, and there are far more than that all over the world. And this is largely a result of acid mine drainage. So what is acid mine drainage? Well, it occurs when mines are abandoned, and those abandoned mines are exposed to the elements like air, oxygen, and water. And chemical reactions ensue from this environmental impact on the abandoned mine. Now, these chemical reactions ultimately lead to acid water being produced, which will flow by over nearby rocks, leaching out metals into the water system, and then the water will flow into the watershed. So now we have toxic water being put into our nearby aquatic ecosystems, and our watershed, our drinking water, all the other commercial uses of water have now been contaminated by toxic metals. This cannot be fixed. It can only be rectified. We have systems that are going to help clean the water because we can't just undo the damage that's already occurred. We must actively try to just keep up with the contamination in our ecosystems. So there are two types of remediation systems. There are active systems and passive systems. Active systems require a lot of maintenance. They're more expensive, and it's basically putting chemicals into the system to clean the water. Passive remediation systems are far less expensive. They use natural remedies to be able to clean the water, and they don't require as much maintenance as the active systems do. Now, the type of remediation that occurs in these systems is discharge dependent, meaning the type of contamination is going to cause us to tweak the type of system that is going to try to fix this contamination. For example, if we have acid in this acid water that we're trying to contaminated acid water we're trying to remediate, our system is going to have a limestone channel in it that is going to raise that pH. Whereas if we had the contamination that wasn't acidic, if it was circumneutral, we're not going to have those limestone channels. It's going to raise that pH because it's already at a higher pH. So the system we're going to be looking at in particular is Middle Branch. It's an acidic system, and its primary constituent is manganese, meaning this system treats manganese and acid. And it's pretty successful at doing that. If you look in the picture below, the white dot represents where the contaminated water is going to start flowing in. The blue arrows represent the flow of the water through each of the ponds until it reaches pond 7, where the last arrow represents it flowing into the nearby creek or lake. And this water is now contaminant-free, and the system has done its job. Recent studies, however, have shown a problem with the system in that Pond 4 specifically is showing high increases of manganese resolubilization. Now, manganese that has been resolubilized is reduced manganese, which is toxic manganese, versus oxidized manganese. Now, we are relating this to ponds 2 and 3 being layered with compost material that encourages for bacterial growth, meaning that ponds 2 and 3 may be encouraging growth of bacteria that are causing this manganese resolubilization downstream in the system. So when we tested the manganese levels across all of the ponds, across all seasons, we saw that ponds 1, 2, and 3 look like they're doing their job. However, we see that spike in ponds 4 and 5 across all of the seasons, leading us to believe this is a biotic feature that is causing this problem. So can we isolate the organisms that are responsible for this manganese resolubilization at Middle Branch? To do this, I took the enriched cultures. I enriched cultures of Middle Branch slurries or water samples, and I enriched them over 4 transfers, and I increased the metal content in each transfer, and I used aluminum and iron to simulate the type of environment that's going to be at Middle Branch. So by the 4th transfer, we had high levels of iron and aluminum in this enriched culture, meaning that only heavy metal bacteria are going to be able to survive in that culture. From there, I placed that enriched culture into sterile pond water and compared the manganese reduction with cultures in the sterile pond water that were not enriched. And what we saw was a fourfold increase of manganese reduction, leading us to believe that heavy metal bacteria in these enriched cultures are the ones responsible and driving for this manganese reduction downstream in the system. So to further find these bacteria that are causing this, I plated that sterile pond water with the enriched cultures onto R2A. I let that grow, and then I isolated and purified some colonies based on their morphologies. I extracted their DNA, and I sent out 4 isolates for shotgun sequencing. Now, when I received the genomes back, I realized that 2 of them were actually duplicates. So today we're going to be talking about 2 isolates that I found at Middle Branch. Phylogenetic trees compare genomes, and they cluster them together to show relatedness. So our isolate CF3R, according to this phylogenetic tree, is most closely related to Ludibacter yeojuensis, but we can also see it's also a close relative of Rhodonobacter. So Ludibacter is commonly associated with cystic fibrosis in the literature. However, it's less commonly associated with contaminated watersheds in the literature, which makes us excited to want to explore that a little bit more. Most interestingly, its close relative Rhodonobacter is known for its nitrate oxidation, and this reaction is coupled with iron oxidation, as you can see on the left diagram. Now, this is super exciting to us because we know that organisms that reduce manganese also reduce iron. And in the study before, when we looked at reduced manganese levels, we did not see any iron reduction. And that was interesting to us because we would have seen that if the bacteria was reducing manganese. So this suggests that we have a symbiotic relationship occurring with our bacteria, CF3R, the ones that are related to this Rhodonobacteria, and the bacteria that are reducing the manganese in the system. This is because once the manganese or iron is being reduced, this symbiotic relationship is allowing the CF3R isolate to oxidize the iron back into the system, but not the manganese. So this is super interesting and fun to understand the symbiotic relationship there. CF7R, according to this tree, is closely related to Burkholderia sapacea. Now, this is a very vast clade, and it's understood in the literature, but it's hard to specifically track down the species based on their reconstruction constantly. So what we do know is that they are present in low nutrient aquatic environments, and that causes problems in the pharmaceutical industry where they have sterile equipment that is used to hold aquatic substances. We see a lot of contamination with this specific clade in those environments. In the literature, we also see a lot of watershed contamination, which makes sense as to why Burkholderia would be found at Middle Branch. When we did a 16S analysis of the relative abundance of bacteria in the enrichment cultures before and after placing them in the sterile pond water, we saw that before we had bacteria of various types, but after we placed them in there, the main bacteria that was present was this clade of Burkholderia, which helps us understand that this is probably why we saw that fourfold increase in the manganese reduction in vitro. So we know that this bacteria, as well as the CF3R isolate, are super interesting as to why they're there, and we want to explore a little bit more about what their role exactly is in the system. So right now, I'm conducting manganese and iron assays to screen for the bacteria that are causing this contamination. I want to further prove that these bacteria are causing manganese reduction, or at least aiding in that manganese reduction, possibly by that symbiotic relationship. Now, by understanding these organisms and their metabolic capabilities, we can begin to understand how to fix the system, such as maybe changing the compost material that's not going to enrich for these specific bacteria that are causing the problems. I want to thank the Center for Undergraduate Research for providing a grant for this project. I also want to thank the CalU Biology Department and also Dr. Michelle Volkanis for her guidance and support during this project. Thank you very much.